I wanted to start by telling you a little bit about my mathematical journey and how I got to study the things that I study. So I am actually from Romania. From a medium-sized city called Galați. So I went to high school there, but I was very fortunate to get a scholarship to Duke, where I was an undergraduate student. And it is at Duke that I studied math as a major, but also that I discovered research and generally discovered applied math and applications to biology. So over here, you have some of the mentors that I still have today. One of them is Tom Witowski, who works in dynamical systems and applied math, and I worked with him on a research project trying to use differential equations to model pendulum systems, like a simple toy problem of understanding how a pendulum goes from taut to slack. And I wrote a thesis with him. And then later on in a different summer, I did research with Mike Reed, who's an established math biologist. He works on mathematical physiology. And with him, I was studying a very interesting question of how the mother and fetus during pregnancy exchange hormones and different kind of proteins, and how that can lead in some women to gestational diabetes. So all of these experiences made me very interested in continuing research and learning more. So I continued with a PhD at Brown, where my advisor was Bjorn Sandstede. He is an applied mathematician and also works in dynamical systems. But this time around, my research also was quite close in collaboration with a biology lab led by Kim Mowry, also at Brown. She works in cell and developmental biology, and I will be telling you more about her research later. But I've basically kind of moved on throughout my career towards more of this interdisciplinary kind of realm, where I really enjoy doing more mathematics, but in support of biology. And so the next step after my PhD was that I did a postdoc. I got postdoctoral training, and I did that at the Mathematical Biosciences Institute. This was at The Ohio State University. And unfortunately, this institute is no longer funded, doesn't exist, but it was a great place for postdocs to essentially, take what they've learned in their PhD, continue some research projects, and become more independent. So I included here on the slide just some people that I worked with while I was at Ohio State, to give you a sense that the work that I've done since has involved teams that are very interdisciplinary. Like some of these people have, appointments in mathematics, but some of them have actually experimental labs in molecular genetics or neuroscience or physics. And I have found this area where it's at the intersection of people's fields to be incredibly exciting to me personally. So hopefully you'll get a sense of that throughout the talk. And finally, if you don't know, I mean, I know some of you are math biologists, but if you don't know so much about math biology, I just wanted to include to tell you that this field, although it's one of the maybe more new fields in mathematics. It has now existed for decades, and it's supported by professional organizations such as Society for Mathematical Biology or the Society for Industrial and Applied Math, which has an activity group on the life sciences. Or, even activity group on dynamical systems has a big focus on math biology. So I just wanted to give you a sense that there's these professional organizations that kind of ground the work of math biologists, and to give you a sense that within math biology, there's a lot of different kind of flavors. In the Society for Math Biology, these are organized as subgroups, which have all these different names. And I've basically highlighted here the ones that I belong to and that I affiliate with most, which are cell and developmental biology and methods for biological modeling or data-driven modeling. But you can see here there's other directions that people take when they work in math biology, like maybe they work in math oncology, that's a huge field, or maybe they work in math epidemiology, and they were very busy during COVID, right, modeling the spread of infectious disease. So to give you a sense that there's different kind of flavors of math biology, and the flavor you'll hear about today will have more to do with these kind of directions. All right, so now to my actual research. So I tend to think a lot about what happens inside of cells. Inside of cells, there's a lot of different kind of proteins that interact with each other and that are very dynamic. And they need to be dynamic in order for that cell to function properly. And so what are some of these proteins? Some of them, I might be calling them cargo. I'm calling them cargo because they're the things that need to be moved inside a cell. So maybe we're inside of neuronal cell and some kind of vesicles need to get to the synapses or they need to be getting to the cell body. Things like that. They could be proteins, they could be RNAs. In this nice video here, you see these very large vesicles. They're a type of cargo. Inside the vesicles, there are other proteins that need to be taken to a certain target destination. So this is the cargo. This is stuff that maybe needs to get somewhere or maybe needs to be uniformly distributed throughout the cell. What actually carries them is basically some kind of vehicle. Like when we go somewhere, we usually take a car or a bike or a bus or something like that. The vehicles for these cargo are something called a motor protein. Um, and these motor proteins essentially walk, in the cells along these tracks that are called microtubules. In this case, is the one that you're seeing here. There's other types of these filaments, but essentially those are kind of like the roads or highways of the cell. So you can imagine really a perfect analogy. It's not so perfect at some point, but an analogy of having highways and roads inside the cell that are these polymers and having the vehicles which are these motor proteins, and having the stuff that is carried like passengers. So I find this kind of fascinating of how this happens, and also how it happens very robustly, despite the fact that inside the cells you have a lot of stochastic fluctuations, like temperature might change. These motor proteins, while they look like they walk very nicely, they really fall off a lot, or there's much more crowded environments than this cartoon shows. And so I'm very interested in how this kind of transport inside of cells happens robustly on time and spatial scales that are observed in experiments. And I'm also interested in other directions of research on how, for example, some of these polymers that form the highways and the roads, basically, how do they get their infrastructure? How do they get really to organize in certain ways? And that organization is ultimately important for both the structure of the cell as well as for the actual transport purpose that they serve. Okay, so I wanted to give you two examples of applications. I will really mostly focus on the one on the left today. But one of the kind of proteins that is transported inside of cells is RNA. And RNA tends to be very important in development. It's important that RNA typically localizes in some area of the cell, whether it is, any kind of cell in your body, or it's an oocyte, an egg cell that is going to develop into an organism, or whether it is a neuron. And so I have two projects that actually have to do with RNA transport, as it turns out. One of them I will talk about more. It has to do with how RNA is being transported in egg cells of the frog. And another one that's a more recent research direction has to do with how RNA is being transported along neurons of mice. Okay, so with that, let's focus on the one that I will tell you about today, which is RNA dynamics in frog development. So I'm going back to Kim Mowry. This is the lab that I collaborated with during my PhD, and I actually still work with them in various directions, continuing this research. Kim's lab is very interested in the fact that egg cells and embryos of different organisms have very distinct spatial features. So you can, for example, see this egg cell or oocyte of the frog, Xenopus levis, having a very clear kind of vegetal side and an animal side. You can kind of think of this as like the yolk and the non-yolk part. That's how it's organized. But this kind of organization is on purpose. It's very important that it is this way. And the reason for that is that if this egg cell then gets fertilized and then cell division happens so that it becomes an embryo, this organization to have kind of an animal vegetal or a top bottom kind of axis to your organism is very important to what those different cells in your embryo are going to do. Like some of them might become part of the bone, some of them might become part of the a muscle, and so on. So they may form different tissues. So this kind of spatial asymmetry creates an axis of growth for the organism, and it turns out to be very important in development. But how does that even come to be? It turns out that RNA, in particular messenger RNA or mRNA, is one of the first indicators of this spatial asymmetry in development. So what you're seeing here is, imagine one of these egg cells over here that we're taking kind of a cross section of, so that we're now seeing a circle, right? And so what you're seeing in the middle is this dark circle, which is the nucleus of the oocyte. And then what you're seeing in bright red is the messenger RNA, this protein inside of the cell that is being fluoresced with a red fluorescent dye. So that way biologists can tell where is the mRNA at different points in time. So early on, you can see it's right under the nucleus, but then later on, it starts being moving and being transported towards the periphery or the bottom of the cell, and then it accumulates more and more there. And it turns out that this kind of accumulation of mRNA at this sort of bottom is very necessary in development. It needs, mRNA has to all get there within 1 to 2 days, but it's not very well understood how that happens. So some research from Kim Maury's lab had suggested in the past that this mRNA transport relies on different kind of mechanisms. But one of the things that it relies on is what that video that I started with, that cartoon video, was telling you about, that the mRNA is probably being moved by some of these motor proteins. There might be different kinds of motor proteins moving it, and they will be moving it along microtubules, like these polymers inside the cells that are kind of long and provide tracks for transport. And so that's basically some of the things that her previous research have revealed. But really, beyond that, we didn't know much more. And so, in terms of coming up with a mathematical model that can be helpful to a biology lab, we wanted to include all the mechanisms that they knew must be there, as well as be able to inspect or try to give some insight into other mechanisms that are harder to do experiments with. And so, for example, in this process, what you're seeing here is that early on in the development of this egg cell of the frog, the mRNA, these red little dots, are basically uniformly spread out throughout the cell. And later on, they start being moved along these microtubules towards the periphery of the cell, and they choose one direction. I can tell you more about that if you're interested. And then they accumulate and accumulate more. And this establishes that top bottom or animal vegetal kind of axis of growth of the organism that is then very important in embryo formation and in forming each tissue with specified kind of cells within each tissues. And behind the scenes, everywhere in these stages here, is the idea that there's probably movement by motor proteins along these microtubules of the mRNA, but there's also probably diffusion because earlier on, everything is uniformly spread out. And diffusion is this physical process that tells particles to essentially fill out the whole space. You can think about a gas that gets into a room and that spreads out everywhere through what's called diffusion. Okay, so somehow there's probably some combination of these two mechanisms happening and maybe more. So let's talk about diffusion a little bit and connect it with how we might model that mathematically. So the way you can imagine diffusion is like in this video over here. See, okay, where the particle is essentially doing these random jumps in different directions, right? And how big those jumps are can be dictated by a parameter, let's call it t, that is the diffusion coefficient. Now, if we wanted to kind of say, what does diffusion do to, for example, some particles that are in 1D in this case, for simplicity of visualization, and where we're starting with all the particles at location 0, what do we expect to happen with the particles if they diffuse kind of randomly? What we expect to happen is that they're basically going to spread out all across that space, right? Remember that analogy with the gas in a room that's going to fill up the whole air, right? So that's what you're seeing here. You're starting with the peak of the particles, and then as time goes on, those particles are filling up all the space. Mathematically, we have an equation that describes this. Now, I imagine a lot of you are studying ordinary differential equations. What I'm going to show you here is something called partial differential equations, but there's nothing to worry about because I will describe to you everything you need to know. Essentially, to describe diffusion, we can think about the concentration of particles that are being diffused, and we can call it V. It's call it V, then the diffusion equation looks something like this. dv dt is equal to d times this operator, right? Like, so second derivatives in space of v with respect to space. Okay, and you can note here how the diffusion coefficient shows up in this equation and tells us how fast really this diffusion process is happening. So there's this basically fairly simple mathematical equations. Now, the reason why this is a partial differential equation, maybe if you're studying ordinary ones, is because we have derivatives with respect to time and derivatives with respect to space in the same equation. But otherwise, things are similar. When it comes to transport, right, because I told you probably these particles are moving by transport on microtubules, and there's biological evidence for this. Well, this video shows what that might look like. It's really just this particle being moved along the microtubules at some speed. Let's call it c. Okay, and so if we wanted to have a similar one-dimensional representation with this concentration of particles at location 0 and trying to understand what happens through time with that concentration, then this would be kind of easy. It would basically just move towards the right with a constant speed. Nothing else would happen to this concentration. It would simply move by transport. Okay, so the transport can also be described by a mathematical equation. It's also a partial differential equation. So now if I'm calling this concentration of particles that are moving, I'm going to call it U. Then I'm going to have this equation du dt is equal to the speed c times du dy. And I'm basically saying everything is moving along dimension y with speed c in some direction. Okay? So we have equations for these two kind of phenomena that we think dictate how the mRNA is moving. Now, if we only had diffusion in this process, and we started with some of these mRNA particles right under the nucleus, as the biologist observed, what would happen after a long time is that the mRNA would kind of fill out the space randomly. Right? So they would be just diffused throughout the cytoplasm of that cell. Now, that's not what happens. There are some oocytes that look like this, where the mRNA is everywhere, but that's really abnormal. That's a mutation. And this would not allow that oocyte to be able to be fertilized and to become an embryo for a future frog. So this definitely is not what's happening fully. Now, if we had transport only, right, these particles would be moving pretty fast, right? They would get to the periphery of this egg cell in less than an hour, assuming they had everything they needed there. They had these tracks, they had these vehicles carrying them. And that's not what's observed either. I was telling you at the beginning that to get all the mRNA where it needs to get in this egg cell, it takes about 1 to 2 days. So neither one of these mechanisms alone can describe what's happening. In reality, what we expect is that it takes 1 to 2 days and the process looks something like this. Okay? So what we do in that case is we can combine our equations that describe diffusion and transport of the mRNA. So I'm going to start with this cartoon here. Here is my concentration U of particles that are being transported. Okay, with speed c, and here is my concentration v of particles that are being diffused. So I'm thinking really of the mRNA that is diffusing as an mRNA that's not attached to any of these motor proteins, any of these vehicles that can carry them. It's just out there in the cytoplasm, and it's moving with these like small stochastic steps throughout space, versus this mRNA in this state, in the state U, is attached to some of these vehicles, is attached to one of these microtubule roads, and it's able to be moved. Okay, so that's kind of the cartoon idea of the model, that we have movement in this population, diffusion in the other population, and then we have these reactions or switching between the two. In other words, with some rate beta 1, we're thinking, well, at some point, these motor proteins are going to unbind from the microtubule, or they're going to reach the end of the microtubule. So then this particle will go from movement to diffusion. And then at some point with some other rate, call it beta 2, this particle that's just diffusing in space is going to find a microtubule, it's going to find a motor protein, and it's going to attach and be able to be moved. Okay? Mathematically, what do? We take those pieces that I introduced earlier and we sort of put them together. So in this concentration U, where we have movement, we have the transport equation that I described to you earlier. And in this V, which is the diffusing population, we have the diffusion equation. If you're studying some partial differential equation, this is also the heat equation. So now I have those two pieces, but I know that there can be switching between them because of what I'm actually modeling. I'm modeling these particles that can attach to a microtubule or unbind from a microtubule with certain rates. And so I add these terms to the equations, which essentially couple the two equations. So now I'm saying at some rate beta 1, some of these particles that are being moved are going to unbind from the microtubule, and they're going to move into a diffusive state. And with some rate beta 2, some of these particles that are diffusing are going to find a microtubule, and they're going to bind to it, and then they're going to start moving. So that's how you have this sort of state switching represented mathematically. Okay, so let me give you a video just like I had of diffusion alone and of transport alone. What would it look like to have both of them together? Well, we would have some transport over there. We had some unbinding and diffusion of the particle. Then the particle found the microtubule again, started moving again, it unbound again, and again goes into diffusion. So you can have, these cycles of going from diffusion to transport and back again, depending on whether you find that microtubule. So those are the equations. And you might notice that one of the things in here is, yeah, we have some concentrations of particles we might care about, but we really also have all of these parameters that we have introduced. I told you about the speed c. That's the speed if a particle is attached to a microtubule, how fast does it move on it? We have a diffusion coefficient d, like how big are really kind of those steps that they're taking when they're randomly moving in the space of the cell. And we also have these beta 1 and beta 2 rates, these rates of attachment and detachment to and from microtubules. So how do we figure those out? This is where I find it really exciting to work with biologists and to work with experimental data to try to back out some of those parameters and understand what's happening. Now, a lot of times in math biology, it's also nice if some of the parameters are known. That turns out to be a little bit hard in the problems I work on because I work with people who work on in vivo or living cells. And in living cells, it's much harder to quantify such parameters. When they do things in vitro on a plate, they might be able to do that a little better and get some of these speeds or speed distributions. But in vivo, in these living systems, it's really hard and their experimental techniques are limited. So let me tell you one experimental technique that they use, which is one of the only ways they have to probe dynamics of proteins inside living cells. This is called FRAP, and it stands for fluorescence recovery after photobleaching. So what biologists, like in the Maori lab, but really a lot of biologists in a lot of different fields use this technique, is they will take some part of a membrane of a cell, and they will fluoresce it, let's say with some orange dye, like you see here. And they will then take a high-powered laser beam and they will bleach some spot, some portion of that region. So that's why it looks dark in here, right? They've just bleached it. And so then what happens next is you expect that some of these fluorescent molecules that are outside the bleached region are going to move in. Some of the bleached molecules are going to move out. And if you wait long enough, things will kind of be well mixed and equilibrate again, right? As you wait through time. But this dynamics in and out of that bleach spot is dictated by what we think is happening there. Diffusion processes, transport processes, whatever we know a priori from the biology that might be happening there. Now, what we actually get from this experiment are these kind of data sets, these time series data sets, these black dots over here, which represent the following thing. They're basically a measure of how much fluorescent molecules we have in a bleach spot. So, for example, at very small times, where, we just bleach that region, we're going to have a very low amount of fluorescence. Like it looks like this should be zero, the way that I drew this cartoon here. In reality, it's biology and nothing is perfect. So there's some small amount of fluorescence still there. Now, at a further point in time, maybe at like 350 seconds or something like that, we are going to see a larger amount of fluorescence in the bleach spot because some of those fluorescent molecules from outside have moved inside. And then as time goes on, sometimes they run these experiments for longer, and this thing will plateau to kind of represent the fact that things have been well mixed. Now, mathematicians have worked, and biophysicists have also worked on trying to model this process that happens in the FRAP experiments, but they've often assumed that the particles simply diffuse or maybe diffuse and bind to some kind of substrate. What we would like to do is we would like to match these kind of experimental curves with curves that come from a model that we believe reflects the biology that we are trying to model. So let's now keep in mind that this curve here, these black dots here, form some function that I'm going to call frap true of time. In other words, it's telling me how much fluorescence I have at each point in time, and it's true because it's what comes from the actual experiment. Now, I told you that I want to model this biological process with this set of equations, right? So the question is, how can I use these equations to extract something that looks like a FRAP curve that I can then try to match with what comes from the experiments? Well, here's, for example, what the FRAP process actually looks like. Here's them fluorescing some part of the cell and then bleaching a circular spot. You can kind of see there. Okay, I just wanted to show you that first. Now, the actual FRAP data, the actual fluorescence in the bleach spot, as it corresponds to our equations, is going to be us taking these two concentrations of mRNA, U and V, adding them together, so U plus V, and integrating them over the bleach spot. So we take the integral over the bleach spot of U plus V, and we expect that to basically be the amount of fluorescence in the bleach spot. Okay, I'm going to call that function frap model of T. It's also a time series because I'm integrating over space, so I'm only left with time. And I'm calling it frap model because it comes from the model. And now let me give you an example of what this process might look like when I model it, right? So this yellow here is me trying to model the fact that we've just fluoresced the whole membrane of the cell. This blue means that we've just bleached some circular spot. This circle is where the measurement is being taken, right, of how much fluorescence there is in the bleach spot. And as time goes on, you can see that if I model these equations, in other words, if I simulate and solve these equations using a computer, what is going to happen is something like what you saw in this video, where that bleach spot kind of moved. There was also diffusion happening and transport as well. And now what I can try to do is I can try to quantify the amount of fluorescence in that bleach spot as time goes on by calculating this quantity. So that might look something like this. I'm basically trying to connect frap true of T, which is the black dots, to frap model of T, which may look something like this red smooth line, because what I'm doing in the model is across, of course, a lot smoother. I'm not adding any stochastic effect in it. So in reality, I'm trying to find this red curve, and I'm really trying to find what parameters C, the speed, D, the diffusion coefficient, and the rates beta 1 and beta 2 would give me a good fit through the data like this. And we don't know ahead of time what those are. And so connecting those two curves essentially means that we set up a loss function where we sum up essentially the difference between the red curve and the black curve at every point in time where we have experimental data. So we're saying frap 2 at time ti minus frap model at time ti with some given parameters. We're squaring that and we're adding all the data from all the time points together to get a loss function. And what we would like for this loss function to do is to be zero. Zero is not really quite possible given that we have, noise from the biological system, but we want it to be as low as possible, right? So we essentially want to find out the parameters in here, those C, D, beta 1, beta 2, that would give us the smallest loss function, basically the best fit through this data. And so this connects to an interesting mathematical field, which is mathematical optimization. And so I've used various techniques from there to try to get at this problem. And so to give you some idea, essentially what we do here is we estimate these parameters by minimizing this loss function. We can also do this for FRAP experiments that are done in different regions of the egg cell. So here's the egg cell again with the nucleus in the middle. And here's different regions where biologists have done this FRAP experiment. And so it turns out that these black dots, like these time series of FRAP recovery, can look very different in different regions of the cell, which suggests that the dynamics is somehow different. Maybe diffusion is more important somewhere, maybe transport is more important somewhere, and so on. And so in our approach, we're able to estimate parameters in these different regions and give insight if they're any different. Um, I will tell you a little bit more about what we find later. But first, I also want to tell you that besides like estimating parameters, we can also do some analysis of the equations. And so if we're going back to this model where we have movement in one population, diffusion in the other population, and the switching between the two of them, it turns out that mathematically it's interesting to study something that's called the effective velocity and effective diffusivity of the cargo. So let me explain to you what that means. It's basically this idea that if I now have the same kind of visualization as I showed you before with space on the x-axis, and I start with the concentration of particles here, and I'm thinking basically of u plus v here. What I expect to see here, if it was just transport, is that it moved to the right. If it was diffusion, I would expect it to flatten out. But if I put both diffusion and transport in there, I might see a combination of the two. I might see some transport to the right as well as some flattening. And that's because I have effects both from the transport with a constant speed and from diffusion, which flattens everything out. So this effective velocity is essentially the speed at which the peak of this concentration has moved, and the effective diffusivity tells us, how this Gaussian distribution essentially kind of flattened. And it turns out that for this simple model that I described to you, we can actually find these quantities, this effective velocity and effective diffusivity. I'm not going to go too in depth on this, but I will tell you that some of this makes intuitive sense. So for example, this effective velocity here, you can see it right here, is equal to the speed c times this fraction, beta 2 over beta 1 plus beta 2. What that means is that we're saying that the speed of the whole population of RNA particles is going to be the speed in the moving population, but it's going to be weighed by a fraction that tells me how many of those particles do I expect at steady state to actually be in a moving state. And that is beta 2 over beta 1 plus beta 2. Okay, so that's why we have this quantity. So that one kind of makes intuitive sense that we cannot have the effective velocity be quite c because only part of the particles are moving, but we expect it to be less than c, and the way that it shows up is exactly like this. Like effective diffusivity is a lot harder for me to explain to you because it has terms that include diffusion coefficient, it has like beta 1, beta 2 in these complicated nonlinear ways, but we have mathematical techniques from dynamical systems that allow us to calculate these by doing analysis of the equations. So I just wanted to give you a flavor of that. I showed you these kind of simple two-state kind of models where the particles can be diffusing and the particles can be moving in one direction. In reality, more complex models may be appropriate for the biological systems that we're studying. For example, in the mRNA movement in frog egg cells, we believe that there are particles that are being moved down by some kind of motor vehicles, and then there are RNA particles that are being moved up by other kind of motor particles. And we know this from the biology. We may still have some of that diffusing population, but we also believe there might be a state where the mRNA is basically kind of stationary. It doesn't look like it's moving at all. It's basically kind of paused. And that could be for various reasons. For example, it's being pulled in both directions by these two kind of vehicles. And so in general, we have tools from both linear algebra and dynamical systems to represent more complex kind of models that have any number of states in them. In this example, we have exactly 4 states. So with that, I told you that I will give you a little bit of an idea of what kind of insights we got by doing that parameter estimation and combining it with some of this analysis. What we have been able to tell biologists is the following thing, that if this is the nucleus of the cell, this is the big, big cell, this is the mRNA. I know these look like bugs. Forgive us for that kind of visualization thing, but this is the mRNA, and these are meant to be kind of the motor proteins. We've given them some insight, some idea that in this upper region, right under the nucleus, there is more kind of transport down. There's more of the motor proteins moving the particles kind of down towards the periphery. As they get closer to the periphery, we see a little bit more of bidirectional transport, a little bit more of diffusion, which is why it probably takes these red particles, these mRNA, 1 to 2 days to fully get there, right? And so this gives them an idea that, the fact that their experimental curves, their FRAP curves looked very different in different regions corresponds to these kind of different mechanisms. Like this one is more dominant here, transport is more dominant here, and diffusion might be more dominant down here. Now, what I showed you so far is this kind of idea represented by the video on the left, where you might have an mRNA kind of moving on a microtubule, diffusing off of it, finding that same microtubule, moving on it again, and then again unbinding from it. And that's maybe appropriate for short experiments. But in reality, what can happen is, of course, that this mRNA diffuses out, but it diffuses far enough that it finds a different microtubule, and it's now going to move on that one. In other words, what I showed you so far are models where the transport was in one dimension only. But really, the transport can be two-dimensional, and it can be driven by this infrastructure of roads and highways that are available, and that they have different orientation. So we have also studied these more complicated settings, right, which are more relevant for understanding things that take a long time, like 1 or 2 days, as this mRNA transport takes. So in order to do that, we have done simulations of the model on basically networks of microtubules that we have modeled in this way. This is an example from the actual biology where they're tagging a protein that makes up the microtubules, so you can kind of get a sense of where the microtubules are. Now they're quite randomly organized, as you can see, but there's a little bit of a radial outward kind of pattern to them. And so that's the kind of thing that we have used to generate these blue microtubules here, which are all model microtubules with lengths that are representative of what the biologist thinks that the length distributions of these things should be. So we create essentially these model microtubules. We then take information from the experiments of what things should look like at the beginning. At the beginning, we should start with the mRNA right under the nucleus. So you're going to see some of these images now where I have the egg cell in my model like this. I have the nucleus in the middle, and I have a lot of the mRNA starting right under the nucleus. That's basically my initial condition for the differential equations that I am going to study. And as I study them, what I'm going to find out is basically things that look like this. So I'm going to simulate those same differential equations with transport in one state and diffusion in the other state and switching between the two. And what I find then, if I start with everything right under the nucleus, is that the mRNA is going to start moving towards the bottom in these kind of distributions. Now, these we were quite happy with because they look fairly similar to what they see in the actual experiment, which you see in this top row. Okay, so we see a similar kind of wedge shape of the mRNA moving towards the bottom in this kind of form. Of course, unlike the biology where they're limited to taking, snapshot at specific times, in this case, 4, 10, and 24 hours, in our simulations, we can look throughout time what's happening. And so we can get a little bit of a better sense of how the mRNA might be moving in the real system. And what you're going to notice here is that I ran the simulation up to 24 hours, so one day, and a lot of the mRNA got to the bottom, but not all of it. And even if I ran it to 2 days, I would notice the same thing, that I don't see basically this complete localization of mRNA at the bottom. And so this kind of timescale suggests that there might be a mechanism we're not including, that we're missing. One of the things that we came up with, and that was based on the fact that some of these things were studied in other model organisms, is that this mRNA might also get anchored in some kind of RNA-dependent way at the periphery. And so we have come up with other types of models where we have studied this both mathematically, analytically, as well as with more complicated numerics to give suggestions of what might be happening at the anchoring site here to keep some of the RNA there throughout time. And that's the kind of very interesting type of thing. Oh, sorry. This is a very interesting type of thing where we can essentially go back to the biologists and give them some suggestions of actual experiments to do. I find this very exciting about math biology is that, we can model a biological, biophysical kind of process, but maybe we come up with something where we can go back and suggest to them, maybe you want to look there. If we don't understand something well enough, maybe this is a place that you can focus your experiments on to try to understand if there's some kind of anchoring protein in here, and that's something that they're doing currently. All right, so with that, I want to also give you just 2 or 3 slides where I give you an idea of some more recent work that we have done for these projects. So it turns out that what I told you so far is that in, this egg cell of the frog, you're going to have mRNA particles kind of moving, diffusing, switching between these states. They've learned a lot more about biology in the meantime in the past few years because their microscopes have gotten better. And so what they've realized is that the mRNAs don't really move by themselves. They tend to organize themselves into something called like granules. They're really called biomolecular condensates, but basically into some kind of clusters that contain both mRNA as well as some other binding proteins that keep them together in that cluster and may stabilize them in those granules. So this is kind of fascinating, and it's very new and kind of revolutionary biology to understand that that is the organization of RNA proteins. And what you're seeing here in the zoomed in version is that these clusters here have RNA, which is in this magenta purple, and they have binding proteins, which is in this green, all together in these clusters. And the clusters, interestingly, are not like vesicles that biologists have studied before that have a clear membrane and everything is shut off. Instead, these clusters are pretty fluid. They can exchange with the exterior pretty readily. They can take up more RNA, they can take up more binding protein, or they can leave some along the way. So these are very interesting structures, and we're very interested in studying how this happens because it's a very different kind of biological organization. Mathematically, we've also worked on some of the parameter inference techniques. What I showed you so far is how can we use things like FRAP data to, get parameters like the speed, the diffusion coefficient, the rates, and then simulate the model with those parameters that we have learned. And this kind of inference techniques have showed us some consistency where the RNA transport behavior, was somewhat consistent in different regions. Like I told you, like in the top, mostly transport, in the bottom, more diffusion or bidirectional transport. But still, within each region, we still had wide variation of the parameters we were getting from different experiments. Like different egg cells, we would get different FRAP curves, we would do the same parameter inference technique, and we would get some variation in certain parameters. Not all of them, but some of them were widely varying. And this suggests something that's called a parameter identifiability issue. Here's what I mean by that. You might have one of these models, like the ones I described to you, with diffusion in one state, transport in one state, and switching between the two. You can put a set of parameters in them, call them parameter set 1. So some speed C, some diffusion D, some beta 1, beta 2, and that will give you a good fit through your data. Then you will take a different set of parameters, call it parameters 2, with different speed C, different diffusion coefficient D, different beta 1 and beta 2, and that fits the data equally well. Like there's not really that much of a difference. And so what that means is that both of those sets of parameters could be viable. They could both give you, fits through your data. And what that means is that you have an issue with identifying the true parameters. You might not be able to identify exactly the parameters, but you might be able to identify combinations of parameters instead. So we have worked on that pretty recently, and we're continuing to work in this direction, trying to say what can actually be learned from certain experiments that biologists do, such as FRAP, and what cannot be learned. And you can only hope to learn maybe combinations of them or something like that. I also wanted to mention that in a different kind of research direction, we study microtubules a little bit more. We focus more on the microtubules rather than, let's say, the RNA or the proteins moving on it. And the reason for that is that these microtubules, I have shown them to you like these tracks, these roads, these highways that are static. Here's where the analogy with our real world life kind of fails, because in fact, in biology, these microtubule roads are dynamic. So they might be like, at one end kind of growing or shrinking and so on. They have dynamics of their own. And this kind of dynamics at both ends allow them to fill out space inside the cell differently and to organize themselves in certain ways that are important for the structure of the cell and also impacts the transport of proteins. So with the different collaborators from Penn State University, we study how microtubules are dynamic and how they organize in neurons of fruit flies, Drosophila fruit flies, which is another very common model organism in biology. So to give you just like a little bit of a sense, they study neurons, right? So you're going to see these longer structures that are either axons or dendrites. In this case, what you'll see in this video is dendrites. And what you'll see is these kind of dots, these dark dots. Those dark dots are going to be dynamic, and what they're going to represent is the end of a microtubule. Turns out the microtubule has a plus and a minus end. We won't get into what those things mean, but essentially this gives you an idea that those microtubule ends are moving. Maybe the microtubule is growing that way, maybe it's moving in a certain way, maybe it's shrinking, and so on. But they are dynamic. And so you can imagine that that can impact how things are being transported as well. And so we have come up with models. In this case, we still use some differential equations for certain things, but we also use stochastic and agent-based models to essentially model these kind of microtubules in dendrites and axons of neurons, and to try to understand how they organize themselves, both in healthy neurons and when they do experiments that cut off the axons and they see some regeneration, and how the microtubule structure has to reorient itself in order to lead to regeneration of that neuron. So there's very interesting questions that have to do not so much with the cargo or RNA or protein transport, but with these actual structure or highways themselves and how they are dynamics and organized. So I wanted to give you a flavor of that as well. Okay, so with that, you may have heard these quotes, but I figured, any math modeling talk maybe should have it. There's this UK statistician called George Box who said that all models are wrong, but some are useful. And he also said, remember, all models are wrong, but the practical question is how wrong do they have to be to not be useful? In other words, we know that models are very simplistic representations of what happens. And that's especially true in biology, where there's so much complexity that we're not modeling in these systems. However, the question is, can we still be useful with these minimal representations that we're using? And I hope that what I showed you here argues that there is use to be made. And with that, some takeaways. Transport inside of cells, I find it a fascinating kind of dynamics. I also find it fascinating that we don't really understand what happens inside of cells and that it motivates interesting new mathematical avenues of research. So I often have to go and learn something new mathematically that I didn't know before because it solves some biological question. I hope you saw that math and mathematical modeling can provide some useful insights into challenging questions in cell and developmental biology, and that math techniques can help us understand specifically mechanisms of different biological processes. So, do we really have diffusion playing a role here? Do we have transport playing a role here? Do we have transport up and down? Like, what is the mechanistic basis of what they see in experiments? Because sometimes they can see things in experiments, but they don't know why they see it, and it's hard to probe experimentally. So again, I also find it really exciting when we're able to suggest new experiments to our collaborators. And something else that I want to say, especially in my field, is that I find that impactful research in this field is often done by teams of researchers. Sometimes they can all be in math, but oftentimes what I find most rewarding is when you have teams of people in different fields like math and biology, physics, biophysics, chemistry, or computer science. And I have worked with people in all of these fields, and I have found it really, really exciting. So with that, there's definitely time for questions and happy to take any.